One of the challenges in AML CAR T-cell development is the lack of really an optimal target. And so the concept in the THINK trial, which will be presented at this meeting, is using CAR-T1, which takes the activating killing receptor of natural killer cells called NKG2D, which is linked to CD3 zeta, and then actually uses the endogenous co-stimulatory molecule DAP10 within the T cell itself. That is sort of the construct utilizing a retroviral vector. So the trial was a traditional dose escalation in patients with hematologic malignancy, did include AML patients as well as high-risk MDS and multiple myeloma, although after dose level 1, all subsequent patients had acute myeloid leukemia. There were 3 dose levels starting at 3 × 10⁸ cells per infusion up to 3 × 10⁹ cells per infusion. As far as safety, which is the primary efficacy for the phase 1 component of the trial, it's been overall safe and well tolerated. We do see cytokine release syndrome occurred in 42% of patients overall, although only 3 out of the 14 patients, or 21%, had grade 3 or 4. The CRS was typically rapidly manageable, frequent utilization of tocilizumab. As far as time to onset of CRS, it's somewhat quicker than traditional CAR therapy, typically an average of 1 day, and really no CRS occurred following 2 days after the infusion. Additionally, the majority were short-lived, so the duration ranged anywhere from 1 to 6 days, with an average between 2 and 3 days, with only 1 patient having CRS lasting longer than that, which was grade 1. Other than that, we have seen hypoxia and pneumonitis in several patients, although that really appears to be sort of in the spectrum of cytokine release syndrome. We have not seen any unique off-target toxicities as far as organ toxicity. Other than that, we do see some worsening of cytopenias, including lymphopenia and thrombocytopenia in some patients. As far as early efficacy, out of the 14 patients, I think encouraging activity to date. So there were 10 AML patients, 8 of which did complete 1 full cycle or 3 infusions of the CAR-T1. Out of those 8 patients, 3 had objective responses, 1 CRh and 2 CRIs. This is by ELN criteria. Additionally, we had several stable disease patients as long as 6 months who did have significant blast reductions, and one of those patients also having hematologic improvement. We only had 2 patients that had progressive disease early on. Both of these patients had a high percentage of bone marrow blasts. We are looking at several translational correlatives. One is looking at NKG2D ligand expression. All patients expressed at least 1 ligand, the majority of which had very high expression, again, of the target of the CAR-T1. We do see chemokine and cytokine induction, but these analyses are ongoing. Additionally, we do see bone marrow tracking of the CAR, which is interesting, and sort of those studies are also ongoing. So in conclusion, CAR-T1, again, it showed encouraging both safety and tolerability, as well as initial activity. Again, this was without standard preconditioning chemotherapy, and with, again, 5 of 8 patients having significant blast reduction. So we are looking to optimize. So one is to potentially decrease the duration or the timing between cycles from 2 weeks down to 1 week. We also have 2 trials, one of which has now opened, one looking called EpiThink, which is with standard flu-cy conditioning. Ideally, that is going to augment the efficacy that we have seen early in our trial, as well as a combination trial with azacitidine. But we are at least hopeful that CAR-T1 will continue to be developed in AML, again, with encouraging data to date.